So the 2024 module 3. Alright, so let's start with divine. The body protects itself from harmful organisms through the actions of the immune system. Define the term immune response. So how would you say it's immune response? Go ahead. Okay, so that little slight raise in tone at the end of the sentence, it seems like you're asking a question. You're asking or you're telling? Mm-hmm. Okay, lovely. Does anybody have another, an alternative response that they would give as well? So that's our okay response. Any other responses? Mm-hmm. 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 You know I say mm-hmm? You know I say no no? Because it's 2 marks. Alright? So that's why I say no no. Oh! Oh, you're just saying it. Okay, lovely. No problem. So, alright, so what are the 5 pathogens then? Pathogens that you know. Okay, sure. Alrighty, so everybody should be okay with our understanding of what the immune response is. If not, I'll just ask a CSEC biology student to enlighten us. Alright? Well, let's look at it, right? So it's the body's defense mechanism against, as we said, pathogens. Right? It's against pathogens. Lovely. Right? It's not really a lot. Right? It's really pathogens or other foreign substances. Because we know that we would have allergens and those type of things, right? Which are not necessarily pathogens. Okay? Substances. Alright, look like we're off to a good start, right? So everybody would generally understand what that is. Everything should be fine. Hold on 1 moment. Alright. So let's go. So suggest 2 reasons why memory cells facilitate long-term immunity. So not divine, but Anushka, can you go ahead? Give me one reason. And because I know Mr. Boville wants to give a reason, Mr. Boville can give me another reason. Alright, so two. So if not, so Mr. Boville, you have to go first before Anushka responds. So give me the first one. Oh, okay. Megan, you want to go ahead? And why does that say Megan? Um, Ariel, is that Shanti McPherson? Anybody else in the call? It's always the 4 people, responding, and the occasional Jonathan. So where's everybody else? Alright, go ahead, Boville. If no one else is responding, then go ahead. Mm-hmm. Give me the reason, then explain. So go ahead. What's the reason? Mm-hmm. Lovely. So what I would state that really and truly as, right, we're talking about a rapid response, not true? Rapid response, right? Upon re-exposure. Rapid response upon re-exposure of a specific allergen, foreign object, or pathogen, right? So any type of thing like that, right? So that's what we're speaking about, right? And that allows or that ties into the second reason now. So we know that the memory cells, memory T cells, memory B cells, right? Which one would actually do the create the antigen rather? Do both memory T and memory B cells create antigens? That's my question. Yes, no, or if you'd like to point out the fallacy. Go ahead, Divine. Well, we need two different. No, this is not two different reasons. This is not. There's another reason. Repeat. Oh, my apologies. My apologies. All right. So we have that. Mm-hmm. Why it sound like you're asking a question? Okay, that's fine. This is the review. Okay. So what's the next thing now? What's the next aspect of the memory cells that facilitate long-term immunity? That is the key to the next reason. To the next obvious reason, anyways. So you know that for biology there can be multiple reasons, but next obvious reason is in that term long term. Lovely. So while the other cells may live for sometimes days, sometimes weeks, depending on conditions, right? The memory T cells can live for many years or even decades, right? So that's one thing we could state. So memory cells persist or exist or thrive or whatever in the body, right? In the body for years. I'll put brackets, right? Even decades. So what you will need to do now is give us our explanation. So since the memory cells produce these antibodies, right, that will allow for rapid response and a more effective response to any type of exposure, right, to any type of pathogen or antigen or well, it's not antigen, pathogen or allergen or anything like that, right? We can tie in now the fact that these cells would live for a very long time in the blood or in the body, right? So since they persist for so long, it provides sustained immunity or protection rather, without requiring repeated infections or vaccinations. We don't require an update for that long, right? So looking at that, providing and sustaining protection without requiring any infections or vaccinations to reproduce any type or rehash any type of antibody production, right? So we're looking at things like that. Anushka, since you never responded, go ahead. Mm-hmm. I remember for persons who are new here, we're not going to write an entire response. Right? We're gonna give the points, we're gonna discuss it. Alright? Answer the question the response to the questions are not what you want. It's how to answer the questions and the information required. So you can answer any question that may come in the future. Alright? Is Anushka here? Lovely Anushka. This is your question. Right? So because we don't this is a virtual platform, we don't really have to create the annotated diagram for now, right? But I want you to describe the entire sequence of events involved in either humoral or cell mediated immunity, right? And because I don't like free choice, this is not a democracy, right? You will give me the humoral response. Alright? So explain to me, give us the steps in the humoral response. Right? This is not a democracy, so humoral, please. Okay, that is fine. That is fine. Anybody want to take the humoral response? No. Why is this meeting so silent? Okay, go ahead. Mm-hmm. Hmm. Hold on. One moment. So speaking about chemical interference using interleukins, right? But I would guess that that would be the second step. What is the first thing that occurs in the immoral response? First thing. Lovely. Mm-hmm. All right. Hold on there for a second. All right. So we have it's responding to the interleukin. All right. So looking at that. So is it so before we go on to plasma cells and stuff like that? That's where you're heading, right? Okay. So let me be very sure. You did mention the release of cytokines to activate B cells, right? Did you mention the release of cytokines in B cell activation? Okay, so let's just do that there. So we just need to make sure that we have everything done. Alright? Lovely. So I'm trying to listen to you. I'm trying to catch anything that is a bit off, right? Or to add in anything that you may have missed, which is fine. Right? So go ahead now. B cell activation. What do the B cells do? Go ahead. Mm-hmm. Mm-hmm. Alright, one moment. Even though I like depth of knowledge, right? We're just going to do an overview, right? So we're not looking at each specific mechanism, right, of antigen neutralization, right? So we'll just look at the antibody production generally, and then we move on. All right. So what's the next step? So after antigen production, is there any other residual step that you'd like to add? I just want to note, I'm not cutting you per se, It's just that that's a lot of information that may make people feel like a bit weird at the moment. So antibody production, that's where you end, and discussing the mechanisms of antibody production. Okay. I think it would be useful after you go through that entire discussion to add a next step or the last step, which is the memory formation. So mention the formation and importance of the memory cells, because in clonal expansion we have plasma and memory, right? So I'd explain what the plasma cells do as you were discussing the secretion of antibodies, right? And then the last thing would have been the creation of the memory cells, or the memory cells being allowed to store these antigens, right, for rapid response upon re-exposure. All right. So that is the case here. And for reviewing, if you're gonna be reviewing this content, I do recommend you look at a diagram while reviewing this content. All right, because some persons can see it while we discuss it, but some persons can't. All right. So Anushka, are you there? Every single question will be given to Anushka until she answers a question. So Anushka, the cell mediated response. So the cell mediated response, Anushka, let's go. It's the first thing that would occur. Alright. So what we're gonna do, we're gonna take 3 steps back. We're gonna take a deep breath. We're gonna ask the invigilator to go to the bathroom. Right? And then we're gonna return to the exam. Alright? So okay, so what do you think is the first thing that should happen? No. 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 Sure. Mm-hmm. Sure. So okay, so anybody else then? Oh, go ahead, Ashley. That's the same thing. Yes, it involves T cells. Yes. So the first thing, before you talk about anything, what must be present for a response to be had? Just a fa- mm, okay, so we're talking about antigen presentation, right? So from phagocytosis, as we stated, right? We know that the phagocyte would present what, the ATP. Well, did I just say ATP? What does it present? The antigen, right? There are two types of cells that normally would undergo the phagocytosis here. So engulfed by which two cells? Not neutrophils per se in this stage, right? But we have macrophage. Well, as it relates to neutrophils, though, no, I don't think the neutrophil would be the best response to this, but dendritic cells would be against a better response. So macrophages and the dendritic cells, right? So, oh, I don't want it to go too far over that, alright? So we have that, the examples of cells that do that, alright? So looking at the APCs, right, being displayed, right, display the pathogen antigens, right? So what APCs displayed, alright, of the antigens. This was exactly what Anushka was saying, by the way, alright? And what else now? Wait, it uses what? It's using the MHC class 1. Class 1 is for infected cells, right? And class 2, right, is for APCs, antigen presenting cells, right? So let me just leave that there, right? But note that class 1 is for the infected cells, and this is using MHC class 2. All right, lovely. Next step, Anushka, go ahead. Next step. Okay, proliferation. So a helper T cells. Well, what do the helper T cells do? They help. Yes, so they would recognize antigens on the MHC class 2, right? And they would release no cytokines. And what do these cytokines do? So cytokines, for example, the interleukin 2, right, would be a cytokine, right? An example of a cytokine. And what does it do? It stimulates. Hold on, let me let this person into the meeting before I continue. All right. So it stimulates what? Oh, well, yes. So it stimulates not only T killer cells but stimulates other immune cells. So this is where the other cells, like the NK cells and other cells, will come into um, action, right? But we're specifically, go ahead. Okay, so question: did the syllabus? I'm not gonna. I'm not gonna. I'm gonna answer your question, no, but I'm asking whether or not the syllabus requires an in-depth understanding of what cytokines are. It was mentioned. All right, so somebody saying something? No. All right, so cytokines, right? Cytokines are what we call signaling proteins. All right. So we remember in Unit 1 when we were discussing the peripheral um, throat membrane cell membrane receptors and stuff like that. How some transmembrane proteins help in cell recognition, right, and cell activation, stuff like that. That's what those transmembrane proteins are, what cells use to communicate, right? So cytokines are one of the signaling proteins rather that will attach to these transmembrane proteins, right, that communicate some information to other cells. So the cytokines would be released by the helper T cells, and you will attach to these transmembrane proteins on T cells and macrophages and other cells, right, allowing them to now act, right? So cytokines are really chemical messages, right? Messages, right? So they're chemical messengers to regulate immune responses, inflammation, and communication. So that's basically it. So you can, mm-mm, not necessarily, right? It's just so the message is a virus or a pathogen is here, right? Cytokine is the envelope that we use to carry that message. All right, so think of it like that. So it carries that message. Lovely. All right. So we have that here. So divine, not divine, Anushka. Well, the helper T cells here, right? But we know that we have cytotoxic T cells. What would those do? Sure. Right. I'll put that there. Right. I have a thing as it relates to terminology. Right. The term is a cytotoxic T cell, but we can call them, I guess, killer cells. The same. The same thing as like saying a red blood cell, but I like to say erythrocyte. Right. But go ahead. hmm Hydrogen peroxide. All right. Okay, lovely. So we're looking at this, right? So one thing, I don't know how important it will be for your exam tomorrow, but it is biology, right? We're looking at the fact that helper T cells would recognize MHC2 complexes, right? Right, or look at it will bind to MHC2 complexes for recognition of the antigen, but cytotoxic T cells look at the MHC1 complex. Right, so that's a subtle difference between the two, right? And we're saying now that they're activated by cytokines from the alpha T cells, right? But what do we say specifically that they do again? We're not gonna look at that yet. We're not gonna look at that yet. Right, we have the proliferation of these two. In three, I would like to speak to the clonal expansion. What does the clonal expansion refer to? Anushka, you're not getting away now. What does clonal expansion refer to? That word that you were having an issue with earlier. Yes, proliferation of the cytotoxic T cells. Lovely, right? So you can either say proliferation of them, or you can say that the cloning, formation of clones, right? That's fine. Four, right? We're looking at the destruction of infected cells, right? Destruction of infected cells. How do we go about that again? So, oh, okay. No, I think no. It's oh, I'm sorry. The way I responded makes it seem like that's not the case, but I think it is indeed the case, right? But hold on. There's something that occurs before that, you know. How we even reaching the cytosol, we have to break the membrane first, not true? Alright? So they release, so look at the release of perforins. Right? So we know that perforins will create pores in the cell, right? So looking at the perforins there, so we know what perforins do. They perforate. That's literally what perforins do. Right? So they would perforate the membrane, so make pores, I guess, in the cell membrane, right? And then we're gonna have the release of what? Okay, we keep on saying hydrogen peroxide, right? There's some so hydrogen peroxide would be involved, right? So looking at the release of hydrogen peroxide H2O2. Sure. So I would call them granzymes, right? So, mhm, right? Or you could just say the chemicals, right? And then you have the example of H2O2 there. And what does that cause? Apoptosis, right? Apoptosis, lovely, right? And then part five now, I always have them in five steps would be go ahead. It's programmed cell death. Yes. Hmm. And then the last step, go ahead. Yes, you could say lysis. Well, as in apolysis, lysis. Hmm. Yeah, lysis can be used. Or we're cutting open the cell, releasing the contents, effectively killing the cell. So yes, right. The last step, I always encourage the last step to be memory. cell formation, so memory T cell formation. Just mention that, right? So go on through both the humoral and cell-mediated response. All right, let's move on with our lives. Let me check the chat first to see if there are any questions, concerns I missed. No? Okay, lovely. So diabetes is a disease in which the body's control mechanism for blood glucose is not functioning well. A study of 20,000 college students was conducted over a 27-year period. Each individual's rate of weight gain and the incidence of diabetes were monitored and recorded. The data was collected in Table 4. We're asked to plot this data, right, as a bar graph with weight in kilograms on the x-axis. All right. So looking at that, are we able to do that? Do we know what a bar graph looks like? A bar graph is not a histogram, right? Let me just state that first, right? Bar graphs are not, all bar graphs are not histograms, right? But all histograms are bar graphs. So something like that. A histogram could be described as a subset of bar graphs, but bar graph not necessarily mean histogram. Just draw a regular bar graph, right? So looking at that, don't draw a histogram to let the assessor guess whether or not histograms identify with being bar graphs. So don't look at that, right? Just draw a regular bar graph, right? That shouldn't be an issue for anybody, right? Nobody should be, putting their hand up and asking, "What's a bar graph?" No, right? Impossible, right? State two key deductions based on the results of this study. So those are the results. What are two key deductions that we can make? All right. So looking at the positive correlation between weight gain and the risk of diabetes. Right? That's what you're saying? Lovely. Between weight gain and the risk of diabetes. All right. What's the next correlation? Well, the next thing that we could state now. Let me just put one there. Next one. Anything else we can deduce? Any other deductions? I'm seeing, well, are we seeing any thresholds? Repeat. To 7.6. Okay, between 6.1 and 7.6, right? So this suggests a threshold effect of some sort, right? So that's what my biology teacher referred to it as, a threshold effect of some sort, right? So we're seeing that the risk sharply rises, right? Or I don't want to say sharply rises. It rises sharply, not true? Sharply. Um, After roughly, let me use the lovely. I don't normally use those signs a lot. Around 6-ish, right? Kg of weight gain, right? So looking at that threshold effect, so this would suggest, right, to the biologist, right, that there's a critical weight threshold for diabetes development. That after you reach around 60 kilograms of weight gain, the risk for developing diabetes skyrockets. So you can see that it has a massive jump. So there's a kind of a threshold effect there. Can we understand what I'm saying? You understand? You sure? Distinction? Okay. State one link between diabetes and diet. Any link. Lovely. So looking at stuff like that. So the increased or the excessive consumption of refined or processed sugars, right? And fats can lead to, it doesn't lead to diabetes, right? It leads to obesity, right? And obesity increases the risk factor for diabetes per se, right? Um, or you could state that, right? So the obesity and what we call insulin resistance, right? Which is a primary cause of which type of diabetes? Which kind? Okay. Mm-hmm. Okay. Type 2 diabetes, right? Look at that. What would be type 1? Okay. Which cell produce insulin in the node of Langerhans? Beta cells. True? True? You sure? I heard somewhere that alpha cells produce insulin. That's not true? Glucagon. I heard from a certain person, Alright, but it's fine. Alrighty, let's go. So now we have the table 5, a heart rate, stroke volume, and cardiac output of two individuals, right? A and B. So lovely. And I really do appreciate experiments like this where you don't identify the person, so the researcher can make any assumptions about the person's right sex or race or anything like that. You just look at clearly what the individual right is experiencing. Right. This is really interesting. So let's have a look. Right. So individual A and B. Right. So we see the table here and we're seeing some filling out needed to be done. Right. So table 5, well, complete table 5 by calculating the missing values. Can we do that? Okay, so that's a clear question, and I'm not answering the question. Now wait for somebody to tell me something. Now wait for you to tell me. Are you sure about it though? Okay, so cardiac output is the product between the heart rate and the stroke volume. So CO, which is the cardiac output, is equal to HR heart rate, right? Times or SV stroke volume, right? So can we just transpose? Is that a possibility? Are we transposing anything or are we just writing? So we can look for individual A first. So what is the cardiac output of individual A? Why am I not hearing the responses? I get nervous when the meat is silent. I get very nervous. Mm-hmm. Yes. Beautiful. Because some people are going to look at the milliliter and do convert to liter because this is biology and not math though. This is biology and not physics. This is biology and not chemistry. That's not how it works, right? So milliliter, so be cognizant of that. So I'm pretty sure that some persons never saw it. Alright? So we must convert milliliter to liter and then do our multiplication. Alright? Now we're going to have to transpose the equation to give us now the heart rate. What would be the transposition? Heart rate is equal to what? Lovely. And what is our response? 70. Lovely. Moving on with our lives. Is then get easy 2 marks? Lovely. Right, now part 2. Identify whether each individual is elite trained, right? Which means performing at least 1 hour of daily intense aerobic activity, or untrained, which implies engaging in moderate aerobic activity occasionally, right? Based on the values given. So, what would you say for individual A? So, individual A has a higher heart rate, right? Lower stroke volume and lower cardiac output. What would you say about A? I'm pretty sure people are typing in the chat, but I'm not hearing anything. So let me look at the chat. Nobody's, guys. Untrained, lovely. All right. What would you say about individual B? Elite trained. Now, at this point within the paper, you stop to look around the entire exam room and wonder, if I did this exam last year, I would have gotten a distinction. Okay, so let's move on. Right, let's move on a little bit now. Part 3. Figure 6 shows how exercise improves cardiovascular health in the human body, right? So we're assuming that exercise could probably improve the health. I don't know. But let's look at it. Discuss how the following improvements in the cardiovascular health shown in Figure 6 impact cardiac output. You must present your responses rather, in complete paragraphs. All right. So what do we say to this now? Go ahead. It is module 3, yes? Alright, hold on. Let's talk about something now. Now, because biology, okay, but because alright, whether it's module 1, module 2, or module 3, I know that the prepared student would be able to answer any question on the paper, right? But the thing is, no, everything is interconnected. So module 2 speaks about human biology and physiology generally, right? Human physiology, etc. Right? But Module 3 discusses its effects on health and the medical application of this biology, right? So everything more or less can tie into Module 2 in a way, right? So, but whether or not you think it's Module 2 or it's more on side of Module 2 or more on side of Module 3, you're able to answer this question, right? The same way how they take Module 2 and Module 3 together to answer this question, we must use the spirit of togetherness to complete these exams, all right? So let's explain this now. Guys, we're doing vasodilation, we're doing increase in mitochondria. Alright? Okay, anybody want to tackle the vasodilation? Mm-hmm. Mm-hmm. Hold on. Hold on right there. Let's nip it right there. Right, one moment. Right. Let me just check something before you continue. It is 8 marks. Alright. So what I would do here for vasodilation, you start with the mechanism of how it occurs, right? Alright. So it says to discuss how the following improvements following improves the cardiovascular health. That's the first part, and then we speak about the impact. That implies that for vasodilation, 2 marks is given to the improvement, 2 marks to the impact. Same thing for increasing mitochondria, 2 marks to movement, to the improvement rather, the mechanism of improvement, and 2 marks to the impact. Right. So I want you to stop you there because we're going into the impact. Right. So let's talk about how it improves first of all. Right? Let's take it in bite-sized pieces and create a cohesive response. So exercise, how? I want you to prove it. Go ahead. I want you to prove it. Yeah, explain. How do they dilate? Lovely. Chemoreceptors. Chemoreceptor. Yeah, baroreceptors and chemoreceptors. Yeah. But in this case, we'll be speaking about chemoreceptors. Alright? Because exercise increases the level of nitric oxide in the blood. Right? Right? By the endothelial cells, causing vasodilation. So I want us to look at the cause. All right. So go ahead now. So as you were saying, Mr. Boville, right? Go into what's happening now. So C2 vasodilation. Go ahead. Hmm. All right. Now I want you to now take us into the impact. So how does that now impact cardiac output? Hmm. How? Hmm. No, I'm doing. There's a reason I'm doing this now, right? Because the thing is, now we don't want to be too vague with the question, right? Vasodilation does not. It okay. Vasodilation itself does not necessarily directly increase blood flow. It improves blood flow. There's something that vasodilation causes that would increase the amount of blood flowing. I want to look at it from that direction. Yes, it does. So looking at the reduced vascular resistance, right, making it easier for the heart to pump. And if it makes it easier for the heart to pump, what's going to be affected, Mr. Bovell? Go ahead. Go ahead. I think was it Divine who was speaking? Oh, go ahead. With the, I'm not hearing the last part of what you said. Oh, if it increases that, well, we don't necessarily have to mention it here per se, but you can, because it could be an impact. All right. But why I'm taking it from this approach is that we can knock out cardio. Some parts of module two and module one is the same thing. But go ahead, Mr. Bovel. How does this impact now? So make the heart easier to pump, right? Easier for the heart to pump blood, right? Then how does this affect stroke volume? It increases stroke volume, true? Okay. All right. Any other thing we could state about this? Any other impact we could state? So that is how it affects the cardiac output, right? So if we speak specifically to cardiac output, that is what we're having here, right? Mm, it can, well, if we have the heart working more efficiently, right? It will set it to the regular beats, no, amount of beats per beat, right? So we don't really increase that there when the athlete is at rest, right? But we're just looking at this, right? Remember, what I'm looking at is in the question it says in figure 6, right? So looking at the improvements in figure 6. Figure 6 also hints to cardiopulmonary oxygen delivery, right? So how would that increase the effectiveness of cardiopulmonary oxygen delivery? So we could just state briefly that better blood flow, right, to muscles, specifically looking at muscles and the cells that use a lot of ATP, right? It really supports aerobic, metabolism, basically, right? So basically, because of the increased stroke volume as well, right? More blood is flowing through the pulmonary artery, right? Allowing for there to be a more effective and efficient gaseous exchange within the alveoli, right? So could speak to that, or alveolar sacs. All right, go ahead. My apologies. Let's go. Hmm. That's true. Yeah, you can note that, right? So they improve the muscle's ability to use oxygen or improve oxygen utilization, right? Delaying fatigue, it actually delays fatigue, right? So could look at that. Nice catch. So delay is fatigue there. Alright. Alrighty. As it relates to the increase in mitochondria, right? The number of mitochondria existing does increase. Alright. I'm not sure how many persons know about the fact of mitochondrial biogenesis, right? But it tends to occur in high performance athletes, right? So looking at endurance exercises would actually stimulate mitochondrial biogenesis, right? So what would be now what's happening here? So we're talking about the exercise. Let's talk about that first. Let me state that first. So it stimulates mitochondrial biogenesis, right? Biogenesis, right? So looking at stuff like that, which is the creation of new mitochondria, right? So what could you say? No, more mitochondria means what? Lovely. More mitochondria equal more ATP. Full stop. We don't have to really go into that, right? So this is really and truly our improvement based off of the question. It's also a thing you need to. I mean, this is something I would tell the unit ones, right? That you need to learn how to basically, or over time, you improve at structuring proper answers for paper twos. But this, for many of you guys here, this is the last leg of exam, so it doesn't really matter by now. Go ahead. Yes, it does. That's what it implies. It directly implies that. All right. So we're gonna end up with faster recovery, right? So when the person has exhausted themselves, right, and they would now need to, have that recovery time, right, as it relates to replenishing ATP stores as well as removing lactic acid, right? And we know that entire process of removing the lactic acid because we know respiration, right? So that recovery time will actually decrease. So recovery will be faster in these athletes because of efficient ATP production. So because we have more mitochondria, right? That means that because we have more ATP production, it helps in sustaining higher output, higher cardiac output, right? So this indirectly, really and truly, helps sustain higher cardiac output. All right. Another thing we could state now, because we have more mitochondria, right? The mitochondria used in respiration, right? We get better. Go ahead. Go ahead. Less oxygen. Oh, that's what I said a while ago. Wait, I'm not hearing what you're saying from you. Okay. Let me stop until I see what you're saying. Let me go into the chat. I think that persons are texting me the chat now. Alright. So Ariel here giving answers. Ariel not opening mind. Yeah, we have less oxygen depth. That's what you're speaking about. Okay, then sure. That's what we're talking about. So since we have more mitochondria, right, we're going to end up with more efficient or better aerobic capacity. Right? So what we're gonna have, is more aerobic respiration and less reliant on anaerobic respiration. Look at that. Should be fine, right? So reduce lactic acid buildup. And we know that already from CSET biology. Right? Anything else? We could speak about a stronger heart, a better heart. Right? So we're gonna have a stronger heart. So the more we do these endurance activities, we'll end up with a stronger heart. Why? Our stronger heart muscles. Even though the heart is really and truly a huge clump of muscles. Right? Why do we have a stronger heart muscle? Because the muscles of the heart now have what? Now have more mitochondria. Right? So since they would have more mitochondria, that would increase seeing what we call contractility as well as stroke volume. And that is it for the 2024 paper. 2024 was not difficult. Alright? That is the paper, guys. That is it. Any questions or concerns?